recorder on this computer. Wonderful. Okay. So let's come back to here. The reason I'm asking you to sit tight is because we're just about to start the light independence, and we can get to the light dependence then a little bit later.
Well, I have a quick question.
Yes, Angela.
Okay, so what you were explaining about the NADP plus turning into the NADPH, I'm interested in how that works with the Kelvin cycle, because in the Kelvin cycle, the NADPH turns into the NADP plus.
Right?
Yeah, so is it because on the chart here, it's like the NADPH is going into the Kelvin cycle and then it's coming out. Is it just like repeating and repeating?
Yeah, so NADPH here, I realize the text box in this isn't great, but NADPH, the reduced version, enters the Kelvin cycle. But then NADPH becomes oxidized, and the oxidized version of that molecule is NADP plus. And that NADP plus then finds its way back to the light dependence, where it'll get reduced again back into NADPH.
Okay.
And we'll see that come up here in a little bit, actually. So I'm going to show you this right now. I'll tell you what is going on here, and then we'll come back to this afterwards to look at it again, because I realize I liked seeing it broken down when I was learning it. This is how I learned it best, is to see each of the stages, but I realize sometimes it's kind of hard to conceptualize the larger idea if we break it down too much. So we'll start by looking at the big idea, kind of talk briefly about what's happening. We'll talk about each step that we see here, 1 through 5, and then we'll come back to this at the end. So the first thing that we're going to look at here is step 1. Step 1 is the light energy hitting the photosystems here. And I'm specifically only going to point out the light hitting photosystem 2. This is photosystem 2, the one that the arrow is pointing to. Even though it's the first one we're going to look at, it's photosystem 2 because it was the second one that was identified. So photosystem 2 is the first one. Photosystem 1 is the second one. So light energizes electrons in the photosystem. It hits the chlorophyll. That's what the green little balls here are. Energizes electrons in those chlorophyll pigments, and then those, because they're super high energy, are able to bounce around and find their way ultimately to some electron acceptors here. That's step 1. Step 2 now is water ends up getting oxidized and donating electrons into photosystem 2, which then gets split into an oxygen and a couple protons, and that's called photolysis or photolysis. Step 3 is electron transport chain. The electrons are being transported from photosystem 2 to photosystem 1. While that's happening, it's actually also pumping protons in as a form of active transport. So we're pumping some protons in a form of active transport during the ETC. Step 4 is, the box 4 is here, but really this is all kind of step 4. Step 4 is more activation of electrons by the light, photoactivation in photosystem 1. And so these electrons that have been energized again, either from the ETC or from photosystem 1, they will start bouncing their way around again up until ultimately this enzyme, which is called NADP plus reductase. If you're looking things up, you might also see the term ferredoxin. You might see ferredoxin as well. This is NADP plus reductase. I refer to it as both in class, but on the test, I would write NADP plus reductase, but in real life, you might also see ferredoxin. So this NADP plus reductase is a wonderfully named enzyme. It's an enzyme, ACE, that reduces NADP plus. And if we're reducing something, what does that mean? If something gets reduced, what does that mean about that something?
Gains electrons.
Exactly. So NADP plus is going to be gaining electrons, and we can see that happening here. If we look in NADP plus, its little electron holders are empty, and in NADPH, they're full. And of course, we have the proton here. So step 4 is the reduction of NADP plus into NADPH. This is the NADPH that we see entering the Calvin cycle. If we go back a couple of slides, if we come here, the NADPH down below is this NADPH. And then finally, step 5 is just a form of passive transport called chemiosmosis. And so it's protons flowing down their concentration gradient through this ATP synthase. This is ATP synthase. And ATP synthase is also wonderfully named. It's an enzyme that synthesizes ATP. We call it phosphorylation. We're adding a phosphate group to ADP, and so we call it phosphorylation of ADP into ATP. So I know it was a lot, it was very quick, but there's the general gist of it. And so we can start breaking it down, looking at it more holistically here. Or I guess not holistically, more individually. So step 1 and step 2 go together very well because they both use the prefix photo. And we all know the prefix photo refers to what?
Light.
Light, exactly. Prefix photo refers to light. So photoactivation electrons means the activation of electrons due to light energy. Step 1 is just the light hitting chlorophyll and the electrons there becoming very high energy. And one of the questions asks, how does this relate to your knowledge of the whatever subatomic particle is getting activated? And so we know that in a molecule or in an atom, we can find protons, and we can find electrons. Electrons are found in these valence shells, right? Don't mind the fact that my electron atom drawing is atrocious. But these valence electrons, or the electrons on the outside, the outermost shell, these are very easy to energize and break off and fall apart. And so that's how, if the question that asks about how does your chemistry knowledge apply to this subatomic particle, that's the idea to think about, is that we're bouncing valence electrons. And so these end up bouncing all the way to this electron acceptor here. Sarah, how you doing?
I'm good. I'm trying to deal with my dog right now.
Okay. Good luck. If you have questions, you can put them in chat or just let me know. So step 1, very simple. Energy from the light excites electrons in chlorophyll, and they bounce around and end up at this electron acceptor. Step 2 is the photolysis of water. So we said prefix photo refers to light. Root word lysis or lyse refers to cutting or breaking. And so the photolysis of water is the cutting of water using light energy. What ends up happening, though, is that the light itself doesn't break the water. What happens is when the electrons trade their energy and bounce from chlorophyll to chlorophyll, photosystem 2 here becomes a wonderful oxidizing agent. It's an oxidizing agent. It's good at oxidizing things, meaning it's good at making things lose electrons. Something like water will end up losing its electrons. So the photolysis of water is water donating electrons to photosystem 2 due to the energy that was provided from the light in step 1. When that happens, electrons are moving into photosystem 2, and additionally, the single the single atom of oxygen is now ready to make a bond to another atom of oxygen to make an oxygen gas. And 2 protons are added into the thylakoid space. This is the thylakoid space. Everyone on board so far?
Yep.
Yeah.
I have a quick question. So when you break water, you get oxygen and hydrogen. What happens to the hydrogen?
That's a great question. Let's come back to that in a couple of steps. Go ahead and ask that in the chat just so we can make sure that we answer it. But we're going to come back to that in approximately 3 steps. So steps 1 and 2, the use of light to energize things and break. Wow, the energize things and break things. Yes, plural. Step 3 now is the electron transport chain, called such because we're transporting electrons in a chain. And where did these electrons in the ETC come from?
Photosystem 2?
Absolutely. All the electrons that got super energized and donated here in photosystem 2, these are the electrons that ultimately end up in the ETC. Again, very aptly named. The ETC is the place where electrons are transported in a chain from carrier to carrier to carrier. However, I have this broken up into step 3 and step 3.5 here, because one of these electron transport proteins is actually a proton pump. What happens is, as the electrons are passed through the ETC, their electrochemical energy is used to pump protons into the thylakoid space. Now, they're coming from the stroma, and the stroma has a low concentration of protons, and the thylakoid space, or the lumen, has a high concentration of protons. So if we're pumping something against the concentration gradient, what do we call this?
Active transport?
Exactly. Exactly. This is a form of active transport. I know it doesn't exactly use ATP like we see in sodium potassium pumps, but the fact is something is getting moved against its concentration gradient, and so it has to be a form of active transport. This has to be active transport. We're going from a low concentration to a high concentration. So step 3 is the ETC and the pumping of protons.
Mr. Ferrari, I have a quick question.
Yep.
Is proton pump, is that going to be the name, like the IB is going to call it, or is there a different name for it? Because I watched the crash course like a few nights ago, and it called it like, I think it was talking about this, but it kind of said like cytochrome complex.
Yeah, so cytochrome C, I think, is actually this, if I'm not mistaken.
Okay, I just didn't know what that was.
I would have to double check that. I will look at that very briefly once we finish the light dependence. I'll check real quick before we get into the Kelvin cycle.
For our test, would we have to know the name, or should we just call it the protein pump?
No, we'll just call it a proton pump for now. And that's what I'm going to look at, is to make sure that that there isn't a given name that the IB actually cares about. I don't remember there being a name for this proton pump that the IB cares about, because I honestly couldn't remember the name. I can't remember the name of this right now. Anyway, phosphoquinone, I think. Oh yeah, I think this is phosphoquinone. I think that's cytochrome C. I don't think you have to know the names of the carriers or the protein. Just know that there are some carriers here, and that there's also a proton pump that pumps protons against the gradient. I think that's going to be the big idea that we care about. The fact is, I don't know the name of it now anyway, so I don't think it matters too much. Okay, cool. So step 4, what we've done so far is we've delivered electrons from photosystem 2 to photosystem 1. That's basically all we've done so far, is we've moved electrons from photosystem 2 to photosystem 1. We've also got some protons down here that we can see. That's it. That's all we've done. We've just moved electrons. The reason that we care about moving electrons, though, is because now, again, we have to be able to reduce NADP plus into NADPH. And if we're reducing something, something is reduced. What does that mean?
Add electrons.
Oh, it gains electrons. Reduction is gain. And so that's why this protein is so aptly named. We'll get there. So electrons finish the ETC and end up in photosystem 1. And additionally, because it's a photosystem, it's still absorbing light energy, and that energy is used to help re-energize these electrons. So this is re-energizing from photoactivation. and ultimately they'll bounce around and find their way up to NADP plus reductase. So one thing I'm going to say here is we can say that photosystem 1 reduces NADP plus reductase. We can say that. And the reason I want to point that out is because I'm pretty sure that that was a point last year with some of my juniors. We can confidently say that photosystem 1 reduces NADP plus reductase. Anytime something gives electrons to something, we're saying that it reduces it. So really, this carrier reduces that protein, which reduces that protein, which reduces that protein. And then, like I said, photosystem 1 will technically reduce NADP plus reductase. I'm going to close my window here real quick. It's getting kind of warm. Just opened it, but it's cold outside. All right, so we've reduced NADP plus reductase from photosystem 1. And again, I want to point out how aptly named this enzyme is. It's an enzyme, ACE, that reduces NADP plus. Recall that NADP plus and NADPH are important because those are our electron carriers. NADP plus is our oxidized version, and NADPH is our reduced version. This does not have electrons. This does have electrons. That's supposed to be a W. Right? NADP plus is oxidized. It does not have electrons. NADPH is reduced. It does have electrons. That's what those two questions they were asking underneath the name NADP plus reductase. What is it doing? What is it catalyzing? The addition of electrons to NADP plus. This NADPH now will find its way to the Calvin cycle. So going back to the overview, the NADPH that we just made here in step 4 is this. Cool. Well, we've actually made 2 of the reactants so far. We've made oxygen gas. That came in step 2 during the photolysis of water. And now we've just made NADPH. So we still need to make ATP. Now, Sierra, you asked what happens to the protons? Chemiosmosis happens.
No, I asked about the hydrogen, because like when you break oxygen, you get.
Yeah,
or
you asked
when
about the
you break-
protons.
So hydrogen turns into protons?
Hydrogen is proton. Hydrogen is proton. Right? And the atomic number of an element is representative of how many protons that atom has. So with an atomic number of 1, that tells us that that element has a single proton, and we know that any element with a single proton is a hydrogen atom. So if you see H plus or you see proton, what you're looking at is a hydrogen atom because it is a single proton. Proton is hydrogen, hydrogen is proton. Does that help now? Does that make more sense?
A little bit. It's just confusing because like when we do it in chemistry, it's not protons, it's hydrogen. So it's hard to like make the connection, I guess.
What do you mean it's not?
Like in chemistry, it's not. They're not called protons. I don't know. I can't think of an element as being a proton because it's like made of protons, neutrons, and electrons. So having it just be a proton just doesn't make sense.
That's the thing, is that this is not made of, this is only a single proton. There aren't even any neutrons here, right? If I have if I have an atomic mass of about 1, that tells me that there aren't even any protons, or I'm sorry, aren't even any neutrons. This is literally a single proton. That's all hydrogen, the element, is a single proton. So I will try and refer to it as both as I continue to explain this. But know that if I say proton, I mean hydrogen. If I say hydrogen, I mean proton. Cool. So step 5, we call step 5 the chemiosmosis of protons. The past 4 steps, we've actually created a large number of hydrogen atoms, a large number of protons inside the thylakoid space here. And again, the thylakoid space is identifiable with having a large concentration of hydrogen ions of protons, and the stroma is identifiable by having a low concentration of protons of hydrogen ions. Where did these protons come from again?
The photolysis of H2O.
Where else? I agree, they came from step 2 when we broke down water.
From NADPH.
In step 4, I see NADPH using hydrogen or being added with a proton with a hydrogen atom and then entering the Calvin cycle.
Is
So
it
where else?
from the stroma through the proton pump?
Absolutely. The ETC was also bringing in protons. So step 5, we talk about where did the protons inside the thylakoid space come from? It came from steps 2 and 3. That's where they came from. Now I technically refer to it as 3.5 here, but steps 2 and 3. If you were to look here and you said, if we were to look in this figure, we'd say these protons that we see here are actually a result of step 2 and step 3.
Wait, so is step 3 just regular diffusion, or was that what we said was active transport?
Step 3 is active transport. We see that the stroma has a low proton concentration and the thylakoid has a high proton concentration. And we see protons moving from the stroma into the thylakoid. That means we're moving from a low to a high concentration. We're moving against the gradient, active transport.
So the energy is coming from the high energy of the electrons?
Correct. The passage of electrons here pulls the protons through.
Got it. Thank you.
So step 5, we've created a giant concentration gradient in the thylakoid space. Or I'm sorry, we've created a large concentration in the thylakoid space, which means that we have a very large concentration gradient going this way. This is the direction of our concentration gradient. Then we know in diffusion that molecules will ultimately move down their concentration gradient to even a concentration out. And we see that here. What we see in step 5 is protons moving from the thylakoid space into the stroma. So this is a form of passive transport. This is a form of passive transport. However, we give this passive transport a very special fun name. And don't ask me why, but we give it a fun name called chemiosmosis. Chemiosmosis is defined as the diffusion of ions, like protons here, like hydrogen ions, down their concentration gradient across a semi-permeable membrane or structure. For some reason, it seems that every single time we want to mention a semi-permeable membrane, it gets a fun name because osmosis refers to that too, but it's fine. The idea here, though, is that chemiosmosis is the process that we see here in step 5. This is chemiosmosis. However, chemiosmosis, so I said, is a form of passive transport, but we can actually give it, it's a type of a type of passive transport. I'm curious if we can recognize what type of passive transport it's a type of. Right, so passive transport, passive transport is transport from high concentrations to low concentrations, from high to low. That's not a low. But it could be simple diffusion, where if someone sprays Axe body spray, that does simple diffusion, but it could be facilitated diffusion as well. It could be facilitated diffusion, where it's from a high to low concentration, but usually with the help of a carrier or a channel protein. And that's what we see here. Chemiosmosis is specifically a type of facilitated diffusion. That's all it is. It's facilitated diffusion with a fun name to it. The channel protein in use is ATP synthase. Picture ATP synthase as a dam. A dam works because the water in a river flows into the dam, turns a bunch of turbines to create static electricity, and then flows out. That's what ATP synthase is really doing here. The water, or the protons, are flowing through the dam, and this little ball piece is literally a rotor. This actually turns. This is a piece of a protein that can actually rotate and turn. And that rotate and turning is pretty much exactly like we see in a dam, where it's the turbines getting, charging and creating static electricity. This rotation that we see here is actually what catalyzes the phosphorylation of ADP into ATP.
So where does that rotation get the energy from to do that rotation?
That the protons moving through it turn it. In the same way that water moving through the dam turns the turbine, the protons moving through ATP synthase turn the rotor. And I know this says mitochondrial, so don't worry about the fact it says mitochondrial, because we're going to talk about this next unit. But the point is that we see the same thing happening here, right? In the dam, water is flowing through and it's turning the turbines and it's creating static electricity. In ATP synthase, water is flowing through, it's turning the turbine and creating energy. So step 5 is just the phosphorylation of ADP into ATP via synthase using chemiosmosis. Synthase uses the chemical potential energy of a proton concentration gradient to drive the phosphorylation of ADP into ATP. When I say the term phosphorylation, it's a fancy word for add a phosphate. So ADP gets phosphorylated. We add a phosphate to it. And there's our light dependence. Right there. Step 1, we said, was the photoactivation of electrons in chlorophyll. So the energy of the light excites the electrons here, and they end up trading and bouncing with each other through chlorophyll molecules, all ending up at an electron acceptor. We said when that happens, photosystem 2 here ends up oxidizing water, breaking it into protons or hydrogen ions and oxygen. And we specifically call this photolysis of water because it's actually the energy of the light that's causing it indirectly. Step 3, we said, was actually like 2 different pieces. First thing is the protons that we just delivered, or the protons that we just energized in photosystem 2, they get passed through the electron transport chain. And while that happens, protons are pumped against their gradient from the stroma, the outside space of a chloroplast, into the thylakoid space, which is a high concentration. So we get pumped from the outside space of a chloroplast into the little discs here. Wait for it. The protons are getting pumped from this space into the little discs. That is what step 3 is doing. Step 4, we said, is the reduction of NADP plus into NADPH. The electrons that photosystem 1 receives from the ETC, they're re-energized and ultimately end up being given to NADP plus reductase. Photosystem 1 reduces reductase, and reductase ultimately reduces NADP plus into NADPH. And that NADPH will then ultimately get used in the Calvin cycle. Lastly, through the couple of previous steps, we created a very large concentration gradient of protons from the thigh-liquid space to the stroma. We were pumping some in, we were producing some by splitting water. So we've got a lot of protons here and not a lot outside. Step 5 then uses that concentration gradient. ATP synthase uses the concentration gradient to phosphorylate ADP into ATP, and we call this chemiosmosis. There's our light-dependent reactions.
Sorry. Where does the phosphate come from?
It's just a lone phosphate.
Okay.
Don't forget that what actually happens in the Calvin cycle is when the ATP gets used, we split it into ADP, right? So we have our ATP molecule, right? This is the adenosine and then triphosphate because 1, 2, 3. And when it gets used, it actually just becomes adenosine diphosphate. And then that phosphate is now free floating.
Okay, thank you.
Yep. Miriam, how you doing?
I'm good. I just had a quick question from the study guide. For question 8B, I was confused, like what to do or like how to do it. So could you just explain question 8B?
Sorry, 8B.
Yeah.
The table? Was that the
Yeah.
table? Okay. Can I ask you to hold off for a little bit?
Yeah.
Okay. Okay, cool. Alrighty. So for those of you who were there for my light dependence, all good?
Yeah, it makes much more sense now. Thank you.
Cool, lovely.
Yeah, it makes more sense. I just have a quick question in like step 4. Are there a certain number of electrons needed to reduce NADP plus reductase?
To reduce reductase. So like you're saying how many electrons are given to reductase?
Because like I feel like on the notes there's like it says like 2 electrons.
Mm-hmm.
So 2?
Mm-hmm.
Okay.
Yep. And so this goes back to when we were learning about reducing NADP plus into NADPH, right? So if I have NADP plus and we see it's being given a proton here, and then that's going to create NADPH. And so what's the issue with this equation charge-wise? Are my charges all balanced?
The product side would need 2 protons.
You're saying that if this were the case that you would see this? Is that what you're saying?
Yeah.
Right. But we don't see that. We see an even charge here in our product, which means that we have to also have 2 electrons. Does that make sense, Kyle?
Yeah, thank you. I just didn't know if I was seeing it right.
Cool. So there's our light dependence. There's also what we call non-cyclic phosphorylation. You don't have to spend a lot of time with this one, but basically this says if there's a lot of NADPH, if you're trying to use it up, we can create a bunch of ATP via the ETC repeatedly. Right, because what we see here is that the electrons go from photosystem 1 to an electron acceptor back into the ETC and back to photosystem 1. So basically we're just pumping a bunch of protons in, a whole bunch, to create a large concentration gradient. I believe the other DP1 bio teachers and I agree that this is not an important piece of information, but doesn't hurt to know. And also going back to, I don't remember who it was, but saying do we have to know the names of it? So yeah, so the first one is phosphoquinone. The proton pump is technically called cytochrome C, or the cytochrome complex, but like I said, I don't think that that's necessary information. Wonderful. So I think, personally, I think the light dependent reactions are more intimidating than the light independents. I think the light independents are much more simpler, but I understand that people can get caught up on what's in a name and what's a carbon and all this other fun stuff. However, at the end of the day, the point of the Calvin cycle is to create our glucose. Well, kind of. We create a precursor to glucose, but we create something that's used to create glucose. In this case, we call it a triose phosphate. Triose phosphate because it's a 3-carbon sugar that has some phosphates attached to it. Now, what I want to point out here, as we hop right into it, and actually I'm going to go to the end, is that we might see a bunch of different names for some of these molecules. The quick overview as to what we're going to see is that the first step that we're going to see is called carbon fixation, and that's this part here. This is step 1, carbon fixation. The next step we're going to see we call reduction or regeneration. And you know what I want to do? I'm sorry, it's not regeneration, it's reduction, which is here. And then the third step we see is regeneration, which is here. Let's write an actual 3 there. I want to start with this one. I'm starting with this diagram here because I want the names of the molecules in this diagram are the names that we will be using that you will most likely be seeing. GP and TP are the names of these molecules that you will probably see the most in this class. RUBP won't change much. However, because chemists are jerks, there are like 5 different naming systems. And so we'll see in this diagram that we see something called PGA, which is here, and something called G3P, but this is actually GP here, and this is actually TP down here. So I want to point that out now that we're going to talk about GP and TP. Sarah, what's up? I saw you bump back.
Nothing much. I just needed to know the Calvin cycle, and I already knew the light dependent reactions.
You said you do need the light dependence?
No, I understood them better than the Calvin cycle.
So like I said, Calvin cycle or the light independence, they're the same thing, has 3 main steps. It's got the first step, which is we call carbon fixation. Second step that's called reduction, and the third step that's called regeneration. And they're all very well named. In the first step, we're going to be fixing carbon, or we're going to be attaching a carbon. In the second step, some sort of reduction is going to be happening. And in the third step, we're going to regenerate something that we need. So they're pretty well named. Now I'm going to start with just the general idea of what's happening. I am not going to talk about the number of carbons yet. I want to make sure that we can follow the names of the molecules and where they are, when they're made, what happens to them. Don't worry about the carbon count yet, but we do need to talk about it. Otherwise, the math doesn't add up. So don't worry about the fact that we're adding 3 molecules of carbon dioxide yet. Just recognize that in carbon fixation, the first step, carbon fixation, atmospheric carbon dioxide is attached to a molecule called RUBP. It's attached to a molecule called RUBP. P. So CO2 enters and it's attached to RUBP, and it does so via an enzyme called RUBP carboxylase, which is referred to as Rubisco. So this is Rubisco. Rubisco is an enzyme that attaches a single carbon dioxide to a molecule of RUBP. And now that actually makes a six carbon molecule here first, but that doesn't last very long. This is back to what Ellie's question was. So that pretty much immediately falls apart into what we call a GP. That's carbon fixation. Rubisco adding a carbon to RUBP. Step 2 is called reduction. In step 2, what will happen is that we will reduce GP into TP. And so if GP is getting reduced, what does that mean is happening to it?
It gains electrons.
It's gaining electrons, which means what molecule do we need that we made previously in the light dependence that has electrons?
Hydrogen. Or
We made
NADPH.
We need NADPH. We need our electron carriers. We need our electron taxis here. Don't forget that the point of NADPH is just to deliver electrons for redox reactions. So NADPH needs to come and it reduces GP, it gives it electrons. And so if NADPH is losing electrons, we say it's becoming oxidized. And the oxidized version of NADPH is just that NADP plus. NADP plus. So we see NADPH enter, we see NADP plus leave. Additionally, we also need a little bit of energy here. Don't forget, we also produced some ATP in the light dependence. That is also needed here. And now when ATP gets used, that gets dephosphorylated into adenosine diphosphate and a single phosphate. Congratulations, that was reduction. Again, carbon fixation was step 1. Reduction here is step 2. So in step 1, we added a carbon to RUBP and it resulted in GP. In reduction, GP was given electrons from NADPH and some energy from ATP and was transformed into TP. Again, you might see it as G3P here, but we don't really care about that in this class. We call it TP. Now, remind me again, what is the overall purpose of photosynthesis? Why does a plant do photosynthesis?
Provide energy.
Say it again, Angela.
Provide energy or glucose.
Correct, to make glucose. Well, here's where we're going to see some of that. What's going to end up happening is that a molecule of TP will actually leave the Calvin cycle. And then this will be used in another reaction to end up being produced, or to end up becoming glucose. So we see a molecule of TP leave. But all we have left to do now is to regenerate RUBP. All we have left to do now is regenerate RUBP. That's step 3. In step 3, we see the use of energy in the form of ATP is used to convert TP into RUBP, and that's it. This is the Calvin cycle. However, if you've been kind of keeping an eye on the carbon count, you'd be very confused as to how we went from this molecule to that molecule. So let's figure it out. What am I looking for? I'm looking
Also,
for,
wouldn't there be confusion in regards to like how GP would have like 6 carbons and then TP has 3 or something?
well, it doesn't, because that was your original question. Your first question was, how does the GP come from if we're adding 1 to 5 and we get 3? Right? That was your first question, Allie, when you first joined?
Yeah, so then, but like after that, doesn't it have like 2 groups of 3?
Exactly, right. So let's get into that.
I have a quick question.
Yes, sir.
So in the video that you linked to this, it said that when you added the 1 carbon from CO2 to the RUBP, it became unstable. Why would it become unstable? Like, because I know that's why it breaks into 2, but I don't know like why it would become unstable in the first place.
I forget. I don't know. My guess would be something to do with electrons, because electrons are jerks. Don't fully understand that one. I just know that it's not chemically stable and falls apart. So we need to start with making sure we know how many carbons we're working with at any given time. And so over on the right side here, I'm going to create a cheat sheet. This cheat sheet is going to keep a running list of the molecules that we have and how many carbons they're made of. So the first molecule that we see here is carbon dioxide, CO2. How many carbons is in a single molecule of CO2?
1.
1. It's a 1 carbon molecule. This is going to be our carbon count. Let's spell carbon right. Carbon count. I have 1 carbon in CO2. I'm also told about another molecule called ribulose bisphosphate, or RUBP. This graph down here tells me that RUBP is a 5 carbon molecule. RUBP is a 5 carbon molecule. However, this also tells me that there are 3 molecules of RUBP. So how many carbons are at this point in time right here? At this point in time, how many total carbons do I have where my arrow is pointing?
9.
I have 3 5 carbons, right? It says RUBP is a 5 carbon molecule, and I have 3 of them.
So 15.
I have 15 carbons at this point in time. Okay.
Oh, okay. I completely missed that. Okay, now I understand it.
So if we know in carbon fixation, a single molecule of carbon dioxide is added to a molecule of RUBP, but I have 3 RUBPs, then that means that we must have 3 CO2s added. So at this point in time, immediately after Rubisco does its job, how many carbons do I have?
18.
Correct. Right, Rubisco has added a carbon to 3 different RUBP molecules. However, this is where we make the unstable intermediate, because at this point in time, it's 18 carbons in the form of 3 6 carbon molecules, right? If we have a 5 carbon and we attach a 1 carbon, that would create a 6 carbon. So we have 3 of those, but each of those will break in half. And so that will result in 6 3 carbon molecules called GP. GP is a 3 carbon molecule. 3 carbons. So I had 15 carbons in the form of 3 RUBP. I added 3 1 carbons in the form of carbon dioxide. And that ultimately results in 18 carbons in the form of 6 GPs. Everyone on board so far?
Yeah.
Anyone not on board? So we said in step 2, reduction. It's called reduction because we're going to be reducing GP. We said we have 6 molecules of GP, and each one of them is going to get reduced by an NADPH. So how many NADPHs are actually going to be used here? If I've got 6 molecules of GP, and each one of those 6 needs to get reduced, how many NADPHs?
6.
6. Each NADPH can reduce a single molecule. And so if I need to reduce 6 of them, I need 6 NADPHs, which means I'm also going to be producing 6 NADP pluses. Additionally, each redox reaction in this case is going to require a phosphate group. And so if I have 6 GPs that are getting reduced, so I have 6 NADPHs, how many ATPs do I need?
Six.
6. Now looking here, looking at this point, oh, I got a bunch of other drawings I forgot about. Looking at this drawing here, at any point in time, did I add or subtract anything carbon-wise?
No.
No. So we should expect to see how many carbons at this point in time?
18.
18. And look, we do. It tells us here that we have 6 molecules of a 3-carbon. This 3-carbon has a wonderful name called triose phosphate, TP. TP is a 3-carbon molecule. So let's regroup here real quick. I'm going to hop back to the left side here. We had RUBP, and we see it's a 5-carbon molecule, and we had 3 of them. So we had 15 carbons here. We added 3 CO2s, 1 for each RUBP. So at this point in time, we should have 18 carbons. 3 times 5 is 15, plus 3 is 18. We don't add or subtract any carbons here. And so that means that at this point in time, we should also have 18 carbons. I'm on board with that. Anyone not on board with that? Cool. How you doing, Nisa?
I'm good.
Good. So finally, we can get into our last part of the Calvin cycle, regeneration. The fact is Rubisco is an enzyme, and we know enzymes are substrate specific, which means Rubisco is really, really good at adding carbon to RUBP and not anything else. So we need to get back to RUBP here. Additionally, we also still need to make glucose. Well, that's what the TP is for. We see that one of our TP will exit the cycle here. One of the TP will exit the cycle. And how many carbons is triose phosphate? Triose phosphate, TP, how many carbons in a single molecule?
3.
3.
3. It's right here. 3. So if I'm losing a single TP, that means I'm losing 3 carbons. So how many carbons should I have here now?
15.
Absolutely. Hey, how many carbons did we start with in carbon fixation?
15.
15.
Wow, crazy. Look at that. What we can do now is go from 5 3-carbon molecules and using energy in the form of ATP, create 3 5-carbon molecules. For every molecule of RUBP that we'll produce, we'll need an ATP. Congratulations. There's the Calvin cycle. Now let's just make sure that we're all on board here. We said that the ultimate goal of photosynthesis is to do what?
Create glucose.
Produce glucose. But so far, the only thing that I've seen made in the Calvin cycle is a 3-carbon molecule. So what the heck? What's going on? What can we do?
Split off the carbon.
What do you mean split off the carbon?
To break it off and make it into glucose using TP.
How are you going to take a 3-carbon molecule and make a 6-carbon molecule? Right, because glucose is C6H12O6. So that's a 6-carbon molecule. Glucose is a 6-carbon molecule. So what needs to happen? If I have a single molecule of TP, what needs to happen?
You need another TP.
We need another one. What's going to end up happening is that every 2 runs of the Calvin cycle will produce enough TP to create a glucose molecule. And that also makes sense because if we think about the equation for photosynthesis, the equation for photosynthesis says that we need 6 carbon dioxides. And hey, look, I used 3 and 1. And so if I do it twice, that would give me 6. Crazy. Look at that. There's the Calvin cycle, folks. There's the light independent reactions.
I have a quick question.
Hey, me.
So when you go around the Calvin cycle one time, does like the stuff you create just kind of wait until you come around the second time, or does something happen the first time?
We can kind of think about it as waiting. You're saying like the TP you're thinking about?
Yeah, like to create glucose.
Yeah, I mean, we can kind of say it just kind of waits. I mean, we can just think about any other, enzyme reactions, right? Enzymes work in reactions when the substrates collide properly. And so the TP that's produced here is just now in the stroma, and then we assume that it can make the proper collision at some point with stroma enzymes and other TP molecules to become glucose.
Okay, thank you.
Yep. How are we doing?
I'm kind of confused on like why in the Calvin cycle are things like being reduced, like the NADPH to like turning into NADP plus, and the same goes for ATP. Like I don't really understand why it occurs there.
What do you mean?
Like how come it happens like during the Calvin cycle and it doesn't happen like prior to that? Like I don't really understand how it relates into the entire cycle as a whole, like big picture wise.
In the light dependent reactions, water and light are both used to phosphorylate or energize ADP into ATP and to reduce NADP plus into NADPH. That is the purpose of the light dependent reactions, is to energize or phosphorylate ADP into ATP and reduce NADP plus into NADPH, so that now the Calvin cycle has energy needed and electrons needed for its reactions. And now once NADPH gives its electrons away, it loses its electrons. We say it becomes oxidized into an NADP plus. And when ATP uses its energy, it uses its third phosphate group, it actually breaks down into ADP, it dephosphorylates into ADP, adenosine diphosphate. This is your larger picture. The molecules that the NADPH that we produce here in step 4 goes to the Calvin cycle, and the ATP that we produce here in step 5 goes to the Calvin cycle.
Okay, now I understand it. Okay, thank you.
Yeah. Any other questions regarding these processes as they stand right now? Because I know, Miriam, you still wanted to look at 8B, correct?
Yeah.
Okay. So is there anything else here that we can look at that people have questions on?
I just have a quick thing. So I feel like the Calvin cycle is just like personally really hard to visualize. And I kind of created my own like little mind maps. Do you mind if I send you those in an email, and would you like look over them?
Please do, sure. And if would you be okay then if I share them with the rest of the class? With the
Yeah,
rest
that's
of the
fine. I'll
students?
send it right now.
Sure, yeah.
Ready. Okay. Thank you.
Okay, so if there's nothing else more on the reactions right now, then let's look at 8B. Let's look at 8B. So 8B on the review. Can I see my application? Okay, cool. 8B on the review asks us to think about the things that we need for the reactions to occur and how they might be able to affect the reactions if we don't have them, right? We're looking at how these can be the limiting factors. So remind me again, why is carbon dioxide necessary for photosynthesis? What is it used in? What process is it used in? What does it help create? Where do we see CO2 used in photosynthesis? Folks.
CO2 is used when it enters the Calvin cycle from the atmosphere.
In the Calvin cycle. Okay. So CO2 is in which part of the Calvin cycle? What's the name of that step?
Carbon fixation.
Yep. So it's used in the carbon fixation step of the Calvin cycle. Specifically, or to be more detailed, Rubisco attaches it to RUBP to result in GP molecules. So that's what CO2 is necessary for in photosynthesis. But if we limit that, right, if we limit carbon dioxide, if I have all the RUBP that I need, so I have 3 RUBP, but I have 5 carbon dioxides, well, 3 of them can enter, right? And so we can get 1 run of the Calvin cycle. That's fine. That produces a TP molecule. But now I only have 2 molecules of CO2 left. And now we can't sufficiently produce everything that we need. We can still attach a couple of these carbon dioxides to RUBP, but that would now only result in what would be 12 GP molecules, or 12 it would be 12 total carbons, because we only have 4 molecules of GP, which means we only have 4 molecules of TP, which means if we lose another one, now we only have 3 TP here, which is 9 total carbons. And that's not even enough to make 2 RUBP molecules. That's kind of what we're looking at here. That's what the second part of these tables is asking about. If you limit whatever we're looking at here, how does that affect? Well, it ends up limiting the production of GP because now we can't properly regenerate our UBP. And because we're not producing all the GP that we can, if we're producing ATP and NADPH in the light dependence, like we see here, NADPH would have nothing to reduce. ATP wouldn't have a lot of things to energize, and so those would back up. And ultimately, then we wouldn't have NADP plus to enter the light dependence. We wouldn't have ADP to enter the light dependence. Something like light intensity. Why is light necessary in photosynthesis? What does light do in photosynthesis?
It excites the electrons.
Photoactivation of electrons, photolysis of water. And so if we don't excite the electrons, then we don't even have electrons to go through the ETC, which means we don't have electrons to give to NADP plus reductase. So we can't reduce NADP plus into NADPH. Or we don't have the proton concentration building because, well, we don't have any electrons flowing, which means this is not happening. This pumping isn't happening. And if we don't have enough light energy, then there's not going to be enough oxidation energy here, which means this water isn't getting cut. And now we have no protons down here. And if we don't have protons down here, do we have a concentration gradient for ATP synthase to work? That's a fat now. No electrons to the ETC, so we don't reduce NADP plus into NADPH. We can't oxidize water, we can't cut it, so because we don't have any electrons in the ETC, because we're not breaking water, we don't have a proton concentration gradient, and so we don't synthesize ATP very well. Lastly, temperature. Where do we see temperature used in photosynthesis?
Doesn't temperature affect the rate of activity
Of
reactions?
Because of what? Yes, it does, but what is it actually? What is it affecting? What is it directly affecting that's causing reaction rate affection? What things in photosynthesis would be affected by temperature changes that would ultimately affect rate of reactions?
The enzymes.
Enzymes. Synthase, reductase, RUBP carboxylase, Rubisco. They're all enzymes. And we learned back in Biochem Part 2 that enzymes are just proteins, and proteins have a defined protein structure that can break if we get too hot, if we get too warm. Or we can say if it's too cold, then there's no energy in the system. So maybe the particles aren't moving enough. And so we don't see those proper substrate enzyme collisions happening. And that's why I wanted to highlight these is I wanted you to think exactly about, I wanted you to explicitly think about where we see enzymes being used in the reactions here, because if you just say temperature, well, that doesn't really mean a lot. And so we have to keep in mind what are the more smaller pieces that we care about here, things like reductase and synthase and carboxylase. Does that help, Miriam? Does that make sense?
Yeah, thank you.
Yep. Any other questions then? Do you want me to restate something from before? Do you want me to talk about something here? Would love to. We got half an hour here left. I would love to talk about this.
For number 5, the question asking why the action spectrum is broader than the absorption spectrum, are you able to go over that a little bit?
Sure. So let's start with the basics, just to refresh ourselves. So an absorption spectrum tells us what wavelengths of light are being absorbed. The absorption spectrum that we are looking at here is specifically for chlorophyll, and even it's a special type of chlorophyll. Chlorophyll A and chlorophyll B aren't exactly the same. But again, it's just generally looking at how well the different wavelengths of light are getting absorbed. So we can see here in this pigment of chlorophyll, we can see a lot of absorption here in the blues and blue-greens, and then a decent amount here in the reds. That relates to something called an action spectrum. An action spectrum tells us at whichever wavelengths of light we want to look at, how quickly is photosynthesis happening, how much photosynthesis is being done. And so if we look at the two together, we can kind of see that they relate pretty well here. I see a lot of action being done here, and that makes sense because at those same wavelengths, I see lots of peaks here of absorption. Similarly, I see lots of action happening here in the higher, and sure, a couple of peaks here at the higher wavelengths. So lots of light is being absorbed at the low and the high. Therefore, lots of photosynthesis is happening at the low and the high wavelengths. But the reason why, specifically here in number 5, the action spectrum being broader than the absorption spectrum of chlorophyll is because this action spectrum is dependent on all of these other pigments. Things like carotenoids, things like xanthophylls, and the fact that there are multiple chlorophyll molecules. So each of these will absorb different wavelengths of light, and all of that will be used to create the action spectrum. So we just look at what would be chlorophyll A, which is this one. I only see peak here at about 450 and peak here at about what would be, I don't know, that's like 425 or so, and then here at about 675 or so. But then I also see chlorophyll B, which has a peak at like 615 and a peak at 460. And then the carotenoids that I see at like 480 as well as nowhere else. And so it's the sum of all of these peaks here that work together to create the overall photosynthesis action spectrum. Right. The action spectrum for photosynthesis is broader than the absorption spectrum of only chlorophyll, because chlorophyll is not the only pigment that is used. Does that help, Anissa?
It does, thank you.
Yeah. Anything else, folks?
I have a question going back to the Calvin cycle.
Yes.
So like, I know that ATP in the regeneration phase, I know that ATP changes TP into RUBP, but like how? Do we need to know like how it does it or that it just does it?
No, don't worry about how. That's college level biochemistry.
Okay.
Because in fact, if you go back and look at one of the diagrams here, I forget which one, because there are like 7. One of the diagrams here shows that we're even popping some phosphates off here already, and then we're adding some back on. It's just, don't worry about how, just know that ATP is used in regeneration to go from 5 3 carbons into 3 5 carbons. And that conversely, the energy from ATP is also used in reduction to change GP into TP. As you can even see here, I see 6 ATPs entering and 6 ADPs leaving, but I don't see any phosphates leaving yet. When I see the phosphates leave after the electrons have been added. So don't worry about the actual mechanisms for whatever molecules are adding and subtracting what.